All right, so we are now on session 11, which is circuitry 7. And now we're going basically back towards the retina. Now we're in the retina again. We went from amacrine cells last time. And so now we're in horizontal cells, okay? So let me just see if I can do this. Okay, so again, the whole purpose of this is to do standard OCT. You know, you're gonna be able to name the layers and really not be nervous about it. Hopefully you're not too nervous about it. You can always figure it out. And then we go through this again and I say, okay, you know, it's complicated, it's not understood, break it up into individual pieces, repeat from different angles the way we always do. But now we're focused specifically on horizontal cells. So remember, horizontal cells are in the outer plexiform layer. They're still in the internuclear layer in terms of their nuclei. They deal with lateral inhibition. Do you remember that way back when I talked about lateral inhibition?
Yes.
Okay, good. So they're lateral inhibition. And so if we remember in the darkness, we know that the central photoreceptor is active. And what does that mean? That means it's secreting glutamate. If the central photoreceptor is secreting glutamate, that means that the horizontal cell that's at its party, so to speak, is also going to be active because they're both like, you know, friends with each other and they're at the same party. But then what is the horizontal cell's job? He's to leave the house, go to the next door neighbor and say, you know, do the opposite of what we're doing here. So he's going to inhibit that person because we want our signal to be loud. Okay, so that's what happens in the dark because it's following the central photoreceptor and it flips the surrounding one. In the light, you already know that the central photoreceptor is inhibited in my parlance here. That means that glutamate is no longer being secreted and the horizontal cell thus is quote unquote inhibited because it's following what, you know, the same valence, if you will, of the central photoreceptor, which means that the surrounding photoreceptors are now active because they're doing the opposite. Do you remember this?
Yeah.
Good. So that's what the horizontal cell's job is. It's sort of to kind of assist, right? These are the helper neurons or the support neurons, as I said before, for that, like, you know, group of 5, those 5 neurons that are part of the team, right? Okay. So lateral inhibition, we know this is an interneuron because it's going from one neuron to another, right? So it's going in this particular schema, it's going from one photoreceptor to another photoreceptor. That's how I've drawn it, right? So this is how it goes. Photoreceptor, horizontal cell, photoreceptor. And we know that it's activated by glutamate. But now we're going to introduce just a teensy bit of a little bit more complexity because we're supposed to learn this, right? These are also GABA-containing cells. Now, what is GABA? You remember it's a neurotransmitter. And in your mind, and I know that it's in my mind too, it's an inhibitory neurotransmitter, right? Because that's how we define even our amacrine cells. Do you remember how an amacrine cell could be GABAergic? Do you remember this? Or glycinergic, right? We defined them via their inhibitory neurotransmitter. But just remember, that's amacrine cells. And certainly neurotransmitters are not like just inhibitory or not inhibitory. They sometimes are inhibitory, but they can also be other things, okay? So just remember, horizontal cells have GABA. They're activated by glutamate, but they have GABA. So most people will look and say, okay, well, I know how horizontal cells work. Here's a horizontal cell with my color coding here, this very red kind of thing. And here are there's my central photoreceptor over here. And then I get my surrounding photoreceptors over here. And then I have my, like, you know, my processes that are connected to them. So what would people say is, okay, well, you know, if I'm being activated, so to speak, by my central photoreceptor, then I'm going to go over and inhibit the guys next door, right? So naturally you would say, well, if I'm inhibiting here, this is probably GABA, but the answer is that it's not. Okay. And I just want to stress this because this is a very specific thing about horizontal cells that makes this organ, in my opinion, you know, obviously I'm a retina specialist, so I'm going to say it's like the greatest organ ever, but like it really, really is a cool feature. Okay. So let's dive down into your book and say, okay, they drew this specifically to show you where the horizontal cells actually connect into the cone and into the rod. So here's your horizontal cell. So we've talked about all that other ones, the FBs, the flat bipolar cell. Now this should totally like make sense to you. You're like, okay, I know what the FB is. I know what the FMB is, flat midget bipolar cell. I know what the IMB is, the invaginating midget bipolar cell. And then I look at the H and it says, that's the horizontal cells, but it's within or nearby the synaptic ribbon. Now, we didn't really talk about that outside of just noting that this is the way it's structured, but now we're going to go into why, okay? So those are your horizontal cells, and notice, even in the rod, you have the same situation. You have the horizontal cell diving down deep into the into the photoreceptor, and it's actually like really, really close to the center to this thing called the synaptic ribbon. Now, why is this the case? Why is it that like you have this horizontal cell gets this like weird special placement? Okay, and that's the question you should be always asking yourself. So then you say, well, the reason why is because the way the horizontal cell actually does its communication with the other photoreceptors is through something called haptic communication, which is literally to inject direct current into the photoreceptors themselves. Okay, so here's something called haptic coupling, and I'm just showing you this from Wikipedia. Just know it's a way to actually communicate. Now, you know, like you're in your head, it's like when a neuron communicates with another neuron, we do synapses, right? We have a synapse, we have a neurotransmitter. The neurotransmitter goes and it like diffuses across that like expanse, and then it goes to the other side, and then it's taken up, and then something happens electrically on the other side of it. And that is like most of the time that's standard. That's why we teach that to you. But horizontal cells with photoreceptors do not do this. And why? So if you look here, this is again the synaptic ribbon. Of course, I can always tell you who wrote this. This is a PhD, not an MD, right? Because you've got this like cone and it's like upside down, and the horizontal cells coming in from, you know, below here. And then you have this R, which is for synaptic ribbon. And then you have the cone here, and this is bipolar cell. And then you have this horizontal cell, and it's kind of like showing you all of these like weird channels and stuff like that. And the thing is, it's shooting the electric current directly through plasma membrane channels into that actual photoreceptor. Okay, and it's why does it do this? It's because there is no synaptic delay. It's the fastest inhibition known. Okay, that's what makes the horizontal cell super cool, in my opinion, and maybe one of the coolest cells that exists in our body. Why does it have to do this? Because imagine if you're getting one single photon of light is flipping this one photoreceptor and making it do something different. The horizontal cell's job is to communicate with literally a bunch more photoreceptors next door and flip them all at once to the opposite of what this central photoreceptor does. But if it's doing that with synapses, then that will take it a very long time, relatively speaking. We need that thing to be done on the order of like milliseconds. Right? So you've got to have that inhibition happen ridiculously fast, on par with the photonic transfer. And so this is why the horizontal cell has this special way of doing it, this haptic communication. That's how it's laterally inhibiting these photoreceptors. Does this make sense?
Yeah.
Okay, so that's why it is diving deep into the synaptic, all the way near to the synaptic ribbon, because it needs to get really into that, near to the plasma membrane of the photoreceptor to make this inhibition or whatever it's trying to do. It's got to tell this photoreceptor as quickly as possible what it needs to do so that it's ready for the next photon that comes in. Okay, so that's why it's really important. So then you ask yourself, well, if that's the case, right, that inhibition is done through this interesting, or lateral inhibition as we call it, it's done through this interesting kind of, you know, haptic communication, direct injection of plasma, of current through the plasma membrane, then why does it have GABA? Like, you would think that GABA would be the one to do it. So I always say this part is confusing, and I'm going to apologize in advance. You can already say, you know, if all you say to me is, I know what a horizontal cell does, I figured it out, you know, if all you knew is what I just told you, that's the game, okay? You already know how horizontal cells work. Nobody's going to expect you to know more than that, but I do, right? Because we're not here doing the McNuggets, okay? I want you to know all of this because it actually will make you understand how this circuit works. So we're going to go into what the GABA is for, because the horizontal cell is pretty simple otherwise, okay? So let's go into what the GABA is for. So here's your nice, you know, photoreceptor, three photoreceptor, central, surrounding, and then your horizontal cell. But I'm going to include now our other guys, right? We knew that this play didn't just have, you know, photoreceptors and horizontal cells. There's also this bipolar cell, right? We know this, and you know this bipolar cell, and this is the schema, but let's draw it better to scale. Okay, so it doesn't look like this really. Okay, what it really looks like is this. Okay, the invaginate, let's say this is the cone. Okay, so you also think, I'm going to say that this is an invaginating midget bipolar cell. So first, let's just acknowledge that, you know, there's preferential treatment to this invaginating on bipolar cell. You've seen that before, you know what I'm talking about. And then you get your flat kind of midget bipolar cell, which tends to be off. So now I've just defined, okay, here's your on and here's your off. That's what it is structurally, generally speaking, for these cones. And then here is your horizontal cell. So at a dark state, when we look in the corner here, this is a dark, right? We know that the photoreceptor, I like to say, is like, I'm now like saying it's like sleeping. It's not inhibited, but it's sleeping. But what are the Z's? The Z's are glutamate. Okay. And then what does glutamate do? Glutamate inhibits, and you know this, if we just go through the old schema, glutamate inhibits the bipolar on cell and activates the bipolar off cell while activating the horizontal cell, right? This schema, you know, correct?
Yeah.
Okay, so this makes sense in your head. We've gone over it many times, we've hammered it into your head. So you're like, okay, I got this, right? Okay, now let's do something weird, okay? Because right now, like, I'm looking at these three cells, but this glutamate is what's driving a lot of this. So if I were to just remove, okay, the photoreceptor and just kind of like do something to the horizontal cell, like all I do is put current into the photoreceptor, whether it's dark or light is immaterial at this point. I'm just going to literally put a probe in and stick current directly into the horizontal cell. Okay, so in other words, I am myself activating this horizontal cell. Now, mind you, if I go backwards here, okay, this horizontal cell at this moment is active. So there is a current going through it because glutamate is being secreted and thus it's active. So right now this is active. But if I artificially activate, okay, I'm artificially activating it. I'm not putting light on this. I'm just sticking a current. Here's what you see when you test the bipolar cells. The bipolar off becomes inhibited. There's no glutamate right now. The bipolar off is inhibited, and the bipolar on is active. Now, just if I may, because it's a little confusing, I'm going to go back to this diagram. This is what it normally is, right? When my horizontal cell is active, if you were to look at my bipolar cells next door to him, this is the valence. The off is active and the on is inactive. But if I remove this photoreceptor's kind of tomfoolery, so to speak, and I just activate the bipolar cell just by myself, no light, nothing, all of a sudden the opposite happens, okay? And that's,
The horizontal cell, you mean?
Yeah, so I'm activating the horizontal cell myself. I don't have a photoreceptor secreting glutamate. No glutamate is being secreted. I am just literally sticking a probe in and just saying, I'm going to override your initial feelings on this. There's no glutamate. I know you want to be inactive. I'm going to activate you. And then I'm going to watch what these bipolar cells do. They actually do the opposite of what you'd expect when the photoreceptor was there secreting glutamate. And so you ask yourself, why is this the case? Right? It doesn't necessarily make sense that this would be true. You would think that if I activated this horizontal cell independent of the photoreceptor, it would still act the same way with the bipolar cells. You would still have the same state. Why is it flipped? That doesn't make sense. And so as I example, and why is it doing this? Well, this is what GABA is doing. And now let's get into kind of this. I don't know if it's a classic experiment, but let's look at this experiment. Okay. So here's an experiment that shows exactly what I said. On this top side, you have the horizontal cell. And then you have the bipolar on cell. Okay. And I know that I'm going into these studies, but I want you guys to, not be intimidated by this. Okay. So we're looking at this. It's not actually very difficult to parse. Okay. So you have your horizontal cell here, and we're measuring inside of it, its conduction. So therefore, depolarization is going this way and hyperpolarization goes this way. Okay. So first condition here is light. So this is the one that you canonically talk about. Light means that we're activating and deactivating or doing whatever we want through a photoreceptor. Okay, so if I do light, in this system, the only thing that can respond to light is a photoreceptor. And then what does that do? It turns off the glutamate. So when light is shining, it inactivates the photoreceptor, which is not pictured here. And then what happens to the horizontal cell? He becomes inactive, right? So he depolarizes, hyperpolarizes. Does that make sense?
Yeah.
Okay, but then the bipolar on, of course, we see that bipolar on depolarizing or is active. Okay, this makes sense. But if we then just ourselves put a current, there is no longer the light. We've stopped this, and we're actually doing the current, and we're making the current negative inside the horizontal cell. I'm making the current negative. I'm not having a photoreceptor. All of a sudden, the bipolar on is actually negative as well. It's like following the horizontal cell when there is no photoreceptor involved. And then if I do it the opposite direction, the opposite thing happens. So do you see how this is kind of weird?
Yeah.
Okay, let's do the same thing over here. This is the bipolar off. You have the horizontal cell. For the horizontal cell, when there's a light state, you have glutamate being like stopped at the synapse. So your photoreceptor is inactive, which means it's hyperpolarized, which means that the horizontal cell is hyperpolarized because it's at the same party. And then you have your bipolar off cell that then becomes kind of hyperpolarized as well, because during a light state, the bipolar off cell is inactive. Does that make sense?
Yeah.
Right? So bipolar off is going to be inactive in the light. Okay, now let's do the current. And you say, okay, well, if I do the current, well, if I put a current through the horizontal cell and I make it a negative current, you kind of get this deflection upward initially, like an oppositional effect. This is not as like striking as the bipolar on, but it's still like not like in the way you would expect, right? And then if you do the opposite current with the horizontal cell, you move up here, it actually deflects in the opposite direction, right? So like if you look at it in a light state, it should really follow immediately, but it doesn't do that here. You see what I mean?
Yeah.
So it's like this inversion that happens when the horizontal cell specifically is being stimulated without a photoreceptor around. And this is kind of weird. Like it doesn't necessarily make sense as to why we would need that. So then you say, why is it doing this? And first of all, if you just think about this, I said this was GABA was doing it. And then, you could ask me, you say, Dr. D, well, how can GABA do both? Like, how can it, like you just told me that GABA does this, but I saw the bipolar on cell go one direction and the bipolar off cell go to the other direction. And you said GABA does both of those things? Like, I don't understand, like, how is it doing active, it's activating or it's doing two different things. How is this possible? And so here's one, I thought it was just inhibitory, which it's not, right? It can be many things. It depends on the cell. And so, well, it's not. And so here, let's look at the on bipolar cell dendrite. And this is your on bipolar cell. I want to point out a major difference. So if you look, GABA here is connected, and I know I'm getting into the weeds here, but I like to do this because now you can really see kind of specific weird kind of things that you can start making inferences about, like certain things you can do. So for instance, here's GABA, and it's attached to this chlorine kind of receptor. And when it attaches it, chlorine gets to move through it. Okay. And so in your on bipolar cell dendrite, that's what this means, on bipolar cell dendrite. Okay. And then you have your off bipolar cell dendrite, and you have the same GABA. It says you have a different kind of situation because you have these different channels in these two different on and off bipolar cells. This first channel at baseline has a ton of chlorine inside of it. So when GABA actually comes from the horizontal cell and connects to this guy, chlorine goes out, and that means that you get less negative in the cell, so you get depolarization, which means it's getting activated by GABA, whereas the complete opposite happens here because of this particular channel that is basically pumping out the chlorine, so there's not so much in there, so you get this influx of chlorine. So the chlorine goes in one direction or the other direction precisely because of these two different kind of ion channels that exist for these two different bipolar cells. Does this make sense? You don't have to memorize this. I'm just saying, do you understand how you could have a differential effect?
Right.
Okay. Now I want to point out one last thing. I have told you many times to learn like these cells from different angles. Well, now you know three. If I said, Jeanette, tell me how to identify, just, if you were in like someone, said, hey, in order to save the world, you have to identify whether this is an on bipolar cell or an off bipolar cell. And then you go through it, you say, well, I could do it physiologically. How? I can shine light, like we just talked about. And if I shine light, I know that, one gets activated in the dark state, one gets activated in the light state because of glutamate secretion, blah, blah, blah. That's your standard way. Then you can say, oh, well, I also can tell you, right? If I look at its axonal kind of thing, in the IPL, there's an on and there's an off. You know that spatially. So on pathology, I could do it. But now you have chemically, you could do it because you know that genetically they express two different chlorine channels. right? And so that's cool too. So if, as an example, and I don't think there's a genetic disease like this, but let's say I were to tell you there's a genetic disease that knocks out this particular chlorine, this particular NAK2Cl channel. You'd be like, well, I know that that's going to affect the on bipolar cells, and that's going to be a problem when light strikes this photoreceptor because all of your on bipolar cells are gone. You might say that I have my bipolar cells, but half of its population is, or the ones that are responding to on center are gone. Right? So now you know 3 different ways. I think that's super cool. You can also genetically now say who is expressing which chlorine channel. I know now whether you're on or off. You get it?
Yeah.
Okay. So this is just, I find it cool just because I know it's complex, but it's showing you that there's a method to this madness. And I will explain to you why we're even doing this. So now people are like, when they figured this out, which is, by the way, like, this is not obvious. Okay. They were like, wait, okay, so what's going on here? Like, this is very, it doesn't make much sense. Like, it would be so much easier if just GABA was the inhibitory neurotransmitter to the photoreceptor. That would be easy. Instead, you've got this haptic communication. And then now you're like, the GABA is being used in both activation and inhibition based on these chlorine channels that are in the bipolar on bipolar. What's going on here? Okay. So then you say to yourself, okay, well, let's figure out why is this happening? So here we go, right? Here's the plot. Now we got to parse this line, okay? Okay, so following prolonged background illumination in the mid-mesopic, okay, so basically kind of like, light to the photopic range, which means the light range. So the light is on. I'm going to use that term. The light is on, okay? Mammalian and non-mammalian horizontal cells are depolarized at rest, and then hyperpolarized to brief flashes of light that are greater than the background illumination. Now, let's just parse what the heck that means. Remember this, these horizontal cells, if I'm in the photopic range, okay, photopic, okay, and I have photopic, what does that mean? It means light struck the photoreceptor, correct? So if light strikes the photoreceptor, that means glutamate is not secreted, it's inhibited. So this mammalian horizontal cell should actually become hyperpolarized, right? But here they say they become depolarized if you stay prolonged in illumination. They actually flip their valence. Why do they do that? Because they need to hyperpolarize to brief flashes of light that are brighter than the background illumination. In other words, your horizontal cell is responsible for norming and basically norming your light state. So let's say you turn on the light, you know that you turned on the light, but it would suck if that's all you could do and everything was just constantly on, because then if I turn on the light even brighter than the light right now, you wouldn't see it. Yeah. Something has to norm you back to square one, kind of like understanding that. So how do you do this? Well, the horizontal cell says, well, I'll be the one to do that. I'm going to go back to almost like a dark state, even though we're in a light state. But if I do that, I can't tell the bipolar cells to go back to a dark state, because otherwise then you'll perceive it as being dark. So I need another hand that says to the bipolar cells, hey, I'm going to feed forward and keep you going and keep telling everyone that you're still on, because we're not actually in a dark state, but I need to flip back to dark so that at a brief flash of light, I can flip my valence and do my tomfoolery and inhibit the surrounding photoreceptor and do all that stuff. Does that make sense?
Yeah.
Okay. Isn't it? And so this is where they say this final thing, thus fully light adapted horizontal cells, which means depolarized. That means, okay, if you're light adapted as a horizontal cell, you are now depolarized, meaning you are now basically like you're back to flipping to active as if you're in the dark, right? That's what a horizontal cell is when it's depolarized. It has to flip back to that state so that we can see bright flashes of light. They could tonically release a substance that depolarizes the on-center bipolar, that activates the on bipolar cells and inactivates the off. So that way the brain doesn't think, oh, well, the horizontal cell just flipped back into dark mode. Everyone is dark. Otherwise, when the light goes on, you would be like, oh, great, it's light on. And then all of a sudden you'd think it was dark again. Does that make sense? So what are they doing? That's just called feed forward onto bipolar cells to keep the bipolar cells matching the ambient conditions. Make sense? Mm-hmm. Yeah, Jeanette?
It does.
And I will tell you, this is why I think the horizontal cell is one of the greatest cells in our body, because this stuff is done in like quanta seconds, right? We're talking like light 10 to the minus, I don't know, like I'm going to guess here, like 9, 10 10 to the minus 10 seconds, right? Like we're so small. It's got to do all of this quickly because it needs to be back in its state. So that way you can determine that, oh yeah, I'm at an ambient condition here. And oh wait, there was another flash, like a bomb just went off over there. I need to see that. I need that information. Well, the horizontal cell is doing the heavy lifting for that and doing so in record time. Okay, so that's what makes it cool. Okay, so then I'm just going to show you. You can go through, and I always say, I'm showing you these things. You can always just go through the literature and see if this makes sense. Okay, well, here you go. Mammalian retinal horizontal cells are unconventional GABAergic neurons. Well, that makes sense, right? Because a conventional GABAergic neuron is one that inhibits, right? I mean, we know that. Well, this is not the truth here. So clearly it's weird. Here's another one. Synaptic elements for GABAergic, meaning the GABA feed forward signaling between, a horizontal cell and some bipolar cells are enriched, blah, blah, blah. Okay, but it makes sense that those words would be in the same sentence, okay? And then let's look in one of these, and I'm gonna just read here. Originally, these cells, horizontal cells, were thought to release the inhibitory neurotransmitter GABA onto cones, an assumption supported by reports showing GABA-mediated currents in isolated turtle and cultured porcelain cones. Okay, fine. However, the proposed GABAergic feedback from horizontal cells onto cones did not withstand further functional investigation in primate and other species. Thus, GABA has been largely ruled out as the mediator of horizontal cell feedback onto cones. But you know where the GABA is going. It's going onto the bipolar cells. Make sense?
Yeah.
Kind of, yeah?
Yeah. It's the haptic connection. That's
correct. You got it. It's the ephaptic connections through the synaptic ribbon. That's why it goes in there. That actually is doing the inhibitory or communication with the photoreceptors or the cones, right? But it's not GABA that's doing it. If it was GABA, it would need a synapse and that would take forever. And we would not have that inhibition at the speed that we need it. Okay. So then here's what I like to do because, I just typed in horizontal cells and GABA, and this is why I always say, I caution you from just going and Googling things. Here's what this iWire, I don't know if you ever used wiki.iwire.org, but here it is. And here's what they say. Functional properties of horizontal cells are depolarized by the release of glutamate from photoreceptors, which happens in the absence of light. I agree. Depolarization of a horizontal cell causes it to release the inhibitory neurotransmitter GABA on an adjacent photoreceptor. I do not agree with that.
Yeah.
And neither do you now, right?
Yeah.
And this is what I'm saying, right? Like now you know how this works. The bipolar cells, the horizontal cell is far more complicated and it needs that GABA to match the ambient conditions of bipolar on cells and bipolar off cells while it flips back so it can receive sort of information of another flash in a light setting. Make sense?
Yeah.
Cool. So that's the physiology. I told you that if all you learned was the ephaptic communication, like that's simple, right? But then you've got this extra thing that makes you really understand how it works. And you definitely know GABA is not being used to be an inhibitory neurotransmitter of the photoreceptors. And you definitely know that if you hear that, someone hasn't really read the primary literature on it, okay? So now let's go into just the structure. This is, I promise you, is going to be like fast, but I just want to say, a horizontal cell has little classes too. I mean, they all do. There are 3 types, right? And there's 1, 2, and 3. Thank God. Okay. There's just 1, 2, and 3. And then all of them have, I say this because this sounds silly, but all of them have axons in the human. Why do I say that? And here's one where HC1 means horizontal cell class 1. There's an axon. Class 2, there's an axon. Class 3, there's an axon. Why are we like specifying there's an axon? You might say, Dr. D, I thought, they all have axons. Well, turns out, we're not the only creatures on Earth, right? There are other, species out there. So if you like see here, it says H1 and there's like B type axon bearing. And that's, and then if you look, the cat has 1 in that column, and then it has like 1 in the axon less A type, whatever that is, right? But thankfully humans, all 3 of them are in this axon bearing category writ large. Okay. So we don't have to worry. It has axons as dendrites. Okay. So that's nice. So I always just specify, when I say class 1, it's an H1B type axon bearing, but it doesn't matter. You don't have to know, just it has an axon. It's like a normal, it's like a normal neuron. Okay. So class 1, why do we have class 1 and 2? What's the 1, 2, and 3? What's the difference? This is what we call your classic horizontal cell. It does basically everything. It's small field in the fovea and then has a larger field in the periphery. It typically has like one axon that moves across the OPL. Here's what it kind of looks like. You have this guy over here, H1, and then you follow its axon. It's got one like large axon that's going through the outer plexiform layer. Remember, we're still in the outer plexiform layer. Like nothing has changed anatomically, despite all that weird physiology that I described to you. Nothing has changed. We're still in the outer plexiform layer. And then there's its terminal there, and it goes to rods and cones. Okay, so it's rods and cones. Okay, so it does all types of photoreceptors, rods and cones. Then you have this class 2, which is like a different kind of structure, but it's all cones to all cones. So there are these like specialists. And you just have to know that that exists. I mean, you don't even have to memorize this stuff, but I'm just saying it's more complicated than just a horizontal cell just touches all the photoreceptors. This one specializes in cones to cones, right? So you see cones to cones. And then the third one is like class 1, but a little bit larger. Okay, great. It contacts more cones. Okay, great. But it's like weird. It's like non-S cones to non-S cones. So this is where you always remember. I always tell you about the add-on pack. You know, I use that kind of analogy, right? Like S cones, you know, coniocellular pathway, bistratified neurons, like these are these extra things, blue light, short wavelength, same thing. That kind of paradigm of vision, that add-on ability, that's the add-on pack. So you're going to have these cells that don't even mess with that, right? So in this case, this goes from non-S cones to non-S cones. So it's basically just the medium and long wavelength cones, right? It's doing the red-green kind of one. It's not doing the blues, okay? And it's just, for whatever reason, I really don't understand this, but it's hard to follow this one's axons. Like, I don't quite understand why, but if you read the primary literature, every time they draw it, they draw right here, and then they draw the axon like going off the page, like, because they can't follow it. So I really don't quite understand what the difficulty in the pathology is, but apparently it's really hard to follow these axons, but they have one. So we don't know where they end up, but we know that it's non-S cones and non-S cones. That's all it is, right? So if you look, here's another way of looking at it, kind of like a Christmas tree approach. You have your H1 that here they're just showing you the cones, but it can do all of them. H2 can do all the cones, and H3 only does non-blue cones. And then if you look at it a different way, here's H1 that does both rods and cones. H2 does all the cones, and then H3 does, you know, just the non-blue cones, and then it has an axon that goes nowhere, like, or in question mark, like it's just, that's just the way it is. Okay, so there you go, rods, and then it's a blue cone, and then this guy doesn't do blue cones. Okay, so that's 1, 2, and 3. Okay, so I'm just going to do this. Okay, we're back to this, the old school like way of looking at it, because remember, like, functionally, you know, you just want to remember the generic thing. Okay, so let's just do it again. I'm going to be in the dark state. In the dark state, just to rehash, what happens to the central photoreceptor? Is that active or inactive?
Active.
That's active. And then the bipolar on or the bipolar off is active in this state?
The bipolar off.
Correct. And then the ganglion cell on or the ganglion cell off is active in this state?
Off.
Good. And then the horizontal cell, is that active or inactive in this state?
It's inactive. God, I do this every time.
Yep. Say it again, though. What I always try to say, the horizontal, just at baseline, okay? There's confusion because of that feed forward thing, but don't worry about that. Just say, in the baseline, you said the photoreceptor centrally is active, and this horizontal cell is like wearing the same uniform as that guy.
It's active.
So it's active, and then what happened to the surrounding photoreceptors?
Then they should be inactive.
Correct. So that's your, and then we can replace those words with hyperpolarized, depolarized, blah, 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 right? But you know, this is what it looks like. So let's just do it from the other end, okay? So let's go with light. Light now strikes the central photoreceptor. Is that active or inactive?
No, inactive.
Inactive. Then which one? Bipolar on or bipolar off is active or inactive? Is active. Which one of those?
Bipolar on is active.
Bipolar on is active. And then ganglion on or ganglion off is active if the bipolar on is active?
On.
Ganglion on. And then now in this state is that horizontal cell, which is at the party of the central photoreceptor wearing the same uniform, is that active or inactive?
It's inactive. It's
inactive. And then what happens to the surrounding photoreceptors? Those are active or inactive? What's that? Say that again.
The horizontal cell is not inhibiting the photoreceptor, so they're
correct.
Active.
So therefore they're active. You got it. So it looks like this. You got it. Okay. All right. So now we're to our little, you know, like I said, I always do a little quiz. It's nothing crazy, right? But here's your horizontal cell. Okay. And I'm going to point to a cell and you're going to tell me what neurotransmitter it secretes. Okay. You pretty much only know two, but let's do it. Okay. What neurotransmitter does this one secrete?
It's a cone, so it's glutamate.
Good. Glutamate. Okay. What neurotransmitter does that guy secrete?
The horizontal cell that secretes GABA.
You got it. It secretes GABA, just not in the way that you may think, but it secretes it and just but not at the photoreceptor. Okay. On which cell does this cell's neurotransmitter act? You said this was glutamate. So who gets touched by glutamate?
The bipolar cell.
And who else?
And the horizontal
You
cell.
got it. So all three, right? All three. Why do I ask that? Because here's the next question. On which cells does this cell's neurotransmitter act? You said it was GABA. Who does the GABA affect?
The bipolar cell.
Correct. So only the bipolar cells. Got it?
Yeah.
Cool. Now name the class. So notice what I've done. I have no rods here. Okay.
Yes.
I have no rods here and I have an S cone. So that means it's all the cones, but no rods. So it's all the cones. Do you remember what class it was? I just went through this really quick. Okay.
Class
but like,
2.
yeah, class 2. You got it. Class 2. Okay. How about now? I'm only doing long and medium cones, but I didn't include a rod and it's no, what's that?
3.
Class 3. You got it. And then how about this one? Now it's a rod, and there's a short wavelength, and then there's a innominate cone there, but it's, which one is this one?
So that's class 1.
You got it. Class 1. Exactly. Cool. So our circuit is now complete. This is the whole circuit of the retina. You went through all of it. Okay. So you should be very proud. I mean, this is like, I'm showing you this and you know all of it, right? You see your photoreceptors, you see your horizontal cell, you see your bipolar off and on, you see your, midget ganglion cell, right? And then there's a parvocellular B cell into layer 4C, it's beta, right? You even know that, right? And then you have your amacrine cell helping out, and you know that there's, glycinergic if it's A2 and I had a rod here or whatever. But yeah, so now you know it all. Congratulations, the circuit is complete, okay? So next time is literally, we're just gonna go through the OCT.
Okay.
That's it. Okay. You literally have gone through all the circuits, the entire circuit. Okay. So you should be proud of yourself. This is not an easy slog, but I hope that they're helpful.
This is very, very helpful, Dr. Dio.
I'm very pleased. Okay. So I will see you then, I guess, next week. And then what we'll do is we'll just kind of go through it. I have a lecture but it's just the OCT straight up. Okay.
Okay.
All right. Okay. Bye.
Thanks so much. Bye.